The setup of a large-scale data collection session is a time-consuming procedure that can considerably reduce the remaining microscope time available for tilt series acquisition. This method enables fast setup of large-scale automated tomography data acquisition to maximize the time efficiency of tomography experiments that require a careful selection of acquisition targets. Begin by opening a dummy serial EM instance. Once the first grid square is mapped, use the Serial EM Navigator menu option, Merge, to see the montage in the dummy Serial EM instance. Double-click on the Navigator window to open the grid square map. Search the map and use the dummy Serial EM Navigator option, Add Points, to add image acquisition points on the target of interest. After mapping the new squares, save the Navigator file and merge the Navigator again. Continue until all grid squares are mapped. Again, merge the Navigator file with the dummy Serial EM instance. Run the PyEM Virtual Maps script from the dummy Serial EM menu. Select Tools and select Virtual Anchor Maps. This may take some time, depending on the size and amount.